Hello and welcome, Life Science class. I'm glad we were able to Zoom last week, and we're going to get to be able to Zoom 2 times this week, so I'm excited about that. So let's jump right into today's lesson. We're going to continue talking about genetics. We're wrapping up genetics this week, and then we'll test on it next week. That is the plan. That is the goal. So let's go ahead and jump right in. First, let's review what we talked about last week. Those two, we actually talked about 3 important processes, but I'm going to focus on 2 right now. So we talked about basically kind of the golden rule, or what we would call the central dogma. Oops, that's a highlighter. I want a pen. Perfect. A central dogma of genetics. And what we mean by that is going to be the basic rule that is kind of governing how we get from the information that's coded inside of genes to it actually having a trait or a phenotype, right? So we said that DNA codes for RNA, which codes for protein. So last week we talked about 2 processes that are kind of in between these, kind of symbolizing where these arrows are. The first one, when you're going from DNA to RNA, is going to be transcription. I'm going to put a C there. I'm not going to write the whole thing because I'm going to run out of room. The second process we talked about is translation. So both of those are going to be important to understand as we move into some of the more complex stuff of genetics. So I'm kind of going to introduce some stuff that's, I think, pretty interesting. It's kind of more on the cutting edge of all of this information. And I wanted to start kind of with this recap with talking about how certain types of toxins work. So every now and then in the news, you might hear something about a certain type of toxin called ricin. Ricin is a toxin that's found in castor beans when you isolate them and kind of get just the kind of pure toxin there. And what it does is basically it enters into your cell. So it passes that cell membrane and it does something called inhibits. It's called a ribosome inhibitor, and I forget what the P stands for. I'll have to look that one up. But anyways, it's going to inhibit your ribosomes. Now keep in mind, what do ribosomes do? And what does inhibit mean? Vocab words here. So ribosomes, remember, are going to be what's doing this action. It's what's making your protein. So basically what ricin does is it actually shuts down those ribosomes. It stops them from making in proteins. I'm sorry about my dog. You can hear him crying in the background. That's how much he dislikes this. Let me pause it for a second. All right, I think my dog is done losing his mind a little bit. So let's try that again. So basically what I was saying is ricin is going to be a type of toxin that enters into that cell membrane. So it enters into that cell membrane and it's going to bind to all of those ribosomes that are hanging out there. And remember, ribosomes, or rRNA, as we learned is the fancy name for it, those are what's going to be able to make proteins. Without proteins, we basically, the whole thing will shut down. All of these cells will shut down. They can't make their proteins, they can't send messages, they can't do daily functions. So that's why ricin can be so incredibly deadly, because it enters into the cells and binds with that rRNA, which makes them not able to work. So every now you hear about that in the news. You hear about it with spies a lot for some reason. So spies tend to use it. So the more you know. Now next time you hear about it, you'll know a little bit like, oh yeah, I kind of know a little bit about the science about how that works. So let's move in and talk about mutations and how we've talked a little bit about what they are, but I want to go a little bit more in depth about the types of mutations we find and why they can be, why they work the way they do in terms of transcription and translation. Okay, so we are going to talk about 3 main types of mutations, and we've kind of already talked about how these work a little bit, but I wanted to just kind of reiterate it or just kind of go over it again, just so that you guys can see that not all mutations are the same. They're going to be a little bit different based on where that mutation is occurring and how drastic it is. So the first type of mutation, the one that is probably going to pop in your head the most on what can happen, is something called a base pair substitution. All right, so base pair substitution happens when one part is really affected. How do we want to say that? So let's say one letter or one nucleotide is changed, which affects one part of the protein. So basically, again, at the end of the day, we want to get proteins out of this. Proteins have a specific shape and they look kind of weird. In fact, if you were to Google what proteins look like in terms of kind of like a 3D printed model, they kind of look spindly. They're really weird looking and they have these folds in them. It's kind of like origami. If you know what origami is, it's basically where you have these little pieces of paper and you fold them in specific ways and you can make like animals out of them. I used to be able to make like a frog and like a crane. You guys have probably seen pictures of these, but basically proteins work the same way. So when you change one fold or you change one ingredient of your protein, you can end up with something really, really different. So this first type of mutation, notice what happens here. So this is kind of the example up here. So we have our DNA sequence at the top. We know that it's DNA and not RNA because we have a T there. But what happens in this mutation, in this type of mutation, which is a base pair substitution, only one letter changed. So this A here changed to a C. Here is the ingredients basically for your protein. It has the order of the amino acids because remember, we talked about how every 3 letters is going to be a different ingredient. We use something called a codon chart over here. I talked about this in the video of the last one. But basically, based on each 3 letters, you get a different ingredient. So if I change one of these letters, I get a completely different protein that goes here. So for example, if I'm writing this in sentences, and I know I did a little bit of this with you guys too, but let's say that we have something kind of like this. Let me make a new text box right here. So let's say that my original sequence says, the cat is sad. That makes me sad. But let's say that the cat is sad. In a point mutation is another word for this, or a base pair mutation, we would say the rat is sad. Notice that is still a sentence. It's still kind of similar to what we were saying before, except now we're not talking about a cat, we're talking about a rat. So you're changing the overall meaning of the sentence by only changing one word. You're only changing one little part. So your example for this that you see in humans is going to be sickle cell disease. So my audio deleted, but this is sickle cell disease. You can see it under a microscope right here. You see the red blood cells that are going to be more circular, and then you see the ones that I just circled right here and here. Those are going to be sickle celled, and those are going to cause problems because they can't necessarily get through capillaries and different things like that correctly. And it's one little mutation that causes this, just one letter change, and that entire shape will change. Okay, so the second type of mutation that I want to talk about can be a little bit more, actually not a little bit, a lot more drastic effect on our cell. The second type of mutation that we're going to talk about is called a deletion. Now, deletions happen when some of that DNA segment is deleted, like at least one nucleotide, and it only has to be one. Let's say one little nucleotide is deleted. Then everything after that point in the genome, everything in that DNA segment after it is going to be affected by it. So deletion happens when one or more nucleotides are taken out of a gene, affecting many parts of your protein. And I'm saying parts here in these definitions, but what I really mean is actually amino acids. So amino acids are what we call the little ingredients here that you're seeing there. So we're changing the types of ingredients and we're changing when they happen essentially. So let's look at our DNA segment here. So this is your original at the top like we had before. And you can see your original protein ingredients are listed here. Now let's say I accidentally delete that beginning nucleotide. I delete that base right there. I delete the C, everything shifts down. So that means it not only affects that first ingredient, that first amino acid, it affects that one and 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 that one. So that can have a really, really big, drastic impact on whatever it's producing. So let's look at this in terms of a sentence. If we were to do this in a sentence, let's say the cat is fat and sad. Things are getting worse for our cat. Let's say that I do a deletion kind of where the C is. Let's try that. So let's say the first amino acid is fine, but now I delete that middle, that C right there. Okay, so I still have to have those same breakups of the way the letters go. So let's say I do something like this. Everything shifts down to the point where this is no longer really legible, right? That's not a sentence anymore. When that happens, when everything shifts like that, another term for this, and you might see this on some things if you do some outside research, is called a frame shift, okay? We call this a frame shift mutation because the entire reading frame just changes after you take out that one letter. After I took out that one C, everything changed. It changed the entire sentence to the point where it no longer makes sense. So let's look at a picture. Let me talk about a disease that is caused by this as our example. Okay, so I'm re-recording the audio for this as well because it cut out. But basically what happens here, this is cystic fibrosis. So cystic fibrosis is caused by a frame shift mutation. And what it does is it damages the protein that regulates water and salt intake and basically release from cells. When that gets damaged, mucus can build up in your airways, it can build up in the lungs, and it can also build up in the digestive tract. People who have cystic fibrosis often need lifelong help to be able to regulate this. So you can see in the picture here, you see this is in the lungs taking, so the third type of mutation I want to go over today is something called an insertion. So an insertion mutation is going to be one where you have a nucleotide that is getting basically put into the DNA. So let me kind of just type out a definition to be a little bit quicker. So when one or more nucleotide is put into the DNA or that gene, right? So let's look at what we're talking about here. So notice here I have my original, right? My original gene for this, I believe, I'm not sure what this is for, but it's gene for some sort of protein. And here I have a C and a T between each other. What happens is a little a gets put in between those. So when that happens, that obviously is going to change the protein as well. Now, question for you to see if you were paying attention on the last one. Would we consider this a frame shift mutation? Yes or no? Well, let's look and see. What would this be like if I were to type a sentence? So let's say the cat is mad. It's now moved on to being mad. And I'm going to make an insertion deletion. So I'm going to just enter in a random letter. So X, C, A, T, I, S, M, A, D. All right. Or some, actually it would just end there maybe. So is that a frame shift mutation? Yes. Look, everything after that insertion, everything after that point where I entered the X, everything in that sentence changes onward. So that's where you get your frame shift mutation. Okay? So that's a frame shift mutation when everything changes. Now, your example for a disease that can be an insertion would actually be beta. Let me make sure I spell this right. I always spell this wrong, so bear with me here. So, man, I'm not with the spelling today. Beta thalassemia. So it's a type of anemia that you find with red blood cells where basically hemoglobin is not made correctly. So it ends up with you feeling really tired, really weak. In really severe cases, that can lead to organ damage if there's not correction taken, because basically those cells can't carry iron well, which means that they can't, at the end of the day, transport oxygen as well. So it can be a really debilitating disorder as well that can be caused by an insertion. So that is pretty much it for today. Oh, wait. I'll keep it here. I'll just zoom in on my face since we don't need that here. That's pretty much it for today. Well, actually, you know what? Actually, let me add one more point. And that point is that not all mutations are necessarily going to cause disease. We talked a lot about mutations today that do cause disease, that cause different things like sickle cell anemia and all of these different things. And usually when you think of mutations, you think of that as bad. But in some cases, they end up being good mutations or even just kind of neutral. They don't have an effect. So it's one of those things that it's so crazy to me to think that we have so much DNA, and the fact that so much is able to go right to make you and I walking around and talking and doing all this stuff. It's really kind of crazy. So anyways, mutations are really, they're really rare, and keep in mind that not all mutations are going to be harmful. So most of the ones that we're talking about are harmful because those are the ones that kind of stick in your memory. But again, there's so much genetic information, and there are so many things in place that God has put in place to basically kind of give our genome a little wiggle room, if so to speak. So that's kind of your lesson for the day. I will see you guys tomorrow on Zoom, and yeah, praying for you, miss you guys, and hope to see you guys soon. Bye.